All right, everybody. Welcome back after a good lunch, I hope. And it's my pleasure to introduce Marissa Fahlberg. She's a friend of mine, and she's a flow cytometrist, an immunologist, and entrepreneur with over 15 years of hands-on experience designing, analyzing, troubleshooting flow cytometry experiments. She's passionate about helping scientists generate higher quality, more interpretable data through thoughtful panel design, modern data analysis approaches, and practical innovation. Marissa is the founder of Spotted Tech, where she develops new tools and technologies to make flow cytometry more accessible and reliable, particularly for researchers and communities in underserved regions. So welcome, Marissa, and I'm happy to see what she's going to talk about here today.
Okay. Pointer. Trying to use the pointer. That's it. Control. Yeah. Perfect. Thank you, Matilda. I'm very honored and happy to be here. I'm going to be speaking this afternoon about data visualization and analysis in flow cytometry, something I'm very passionate about. And the opportunity to give this presentation forced me to reflect on all of the lessons and challenges, both philosophical and practical, about data analysis and flow cytometry, and distill it in a way that hopefully will be helpful to people in this room. So without further ado, the builder and the thinker, workflow design, and the search for clarity in cytometry. So the journey of a cytometry scientist, in my experience, I'm going to be projecting here, is long and hard. Cytometry is the marriage. Cytometry itself is a very sophisticated and very complex technology, which marries so many different fields: optics, fluidics, electronics, biology, mathematics, computational science. And that is being used to describe an also extremely sophisticated and complex biological process, the most complex army in the entire world, the immune system. So the task is not trivial. And I think from the presentations this morning, it's a reminder of how challenging this journey really is and continues to be, even for those of us that have been in this field for a really long time. But that is the challenge that all of us in this room have accepted. And so we must start somewhere. And that is the collection of rigorous flow cytometry data. Every cytometrist will always start with something like this, no matter what their presentation is about. You will never regret collecting fantastic data if you put the work in up front. You will save so much time in the end. In the worst case scenario, if your flow data is, in the best case scenario, if your flow data is not great, you will spend a lot of time, labor, confusion trying to analyze it. Your results, at best, may be reproducible. At worst, they will not be reproducible. They will not stand the test of time. And it's just, you're not going to get anything published. So you really have to start with great reproducible data. And our speakers this morning went over some strategies to get that. So after you have gotten your first set of great data, you've submitted all of the correct controls, you're using validated antibodies, you've avoided co-expressed markers, you're using an optimized instrument, you get to step 2, what I'm calling the default settings. So now you're using the default software settings, you're just starting to figure out how to analyze it, so you may also be using default go-to plot types. Like maybe you make a pie chart of the different cell populations, maybe you make a bar chart. You're just happy that the dots are on the plot, they're not doing bananas, they're not going outside your gates. And honestly, this is already a very hard thing to do, and you're already halfway there if you just get to dots that look like that. However, it is also at this stage that you start to read Nature immunology papers, and you see that the flow data that they're publishing and the plots in their papers don't quite look like yours. And so that is when you reach stage 3, what I call overcorrection. Now you are making UMAPs like they are going out of style. You've learned how to use Prism, maybe even R and Python. You're making heat maps and correlograms, and you're so proud of your beautiful UMAP, and you bring it to your colleague and you say, look at what I did. And they say, wow, that's amazing. What does it mean? And you say, I have no idea. Maybe it's just me. I made a lot of UMAPs during this stage. But this is when you move into the final stage of the journey, stage 4, thoughtful communication. Oops. What did I do? Oh. There we go. Thoughtful communication. It's when you realize that if you show your colleague your UMAP and they have no idea that your CD8 T cells went down after your novel treatment was administered, then you're not really communicating your story at all. They're not getting the main idea, which is that your drug, your treatment, your condition showed a remarkable biological difference. And that is the main point. The problem with stage 4 is that you realize, oh, actually one more point. At stage 4, you also start to realize when to use the UMAP and when to use the lowly bar plot, which a lot of people don't like because it starts at 0. But I still kind of like it because it's just one variable at a time and you immediately know the answer. The box plot is technically better, I have to say that in my talk. But it's also at this stage that you start to realize how difficult thoughtful communication is. And that is when we get to part 2 of this talk, the builder and the thinker, the dual mode problem. I realized that I don't think I've ever heard a name given to this, but in my experience, it is extremely difficult to both be the person analyzing the data, especially analyzing very complicated, high-dimensional, sophisticated, using a very wide array of tools and possibilities, and be the same person that steps back and thinks about the big picture and how the results are all connected at the same time. It is very difficult, in my experience, to do both. These are competing mindsets. Data analysis requires meticulous attention to detail, requires understanding math and statistics, correcting small errors, and thinking requires putting it all together, puzzle pieces together. This is not a new concept. It's actually also known in architecture. This guy in the corner, Jean-Baptiste Rondelet, 100 years ago came up with this concept. They had the same problem in architecture, where they had all these architects designing blueprints, handing them to the engineer, who would say, this cannot possibly be built. This doesn't work. And so he always argued that an architect must both be a builder and a thinker. He talked about how hard and challenging that was, but also that it is essential. And so for the next part of the second part, I'm going to try to share a few strategies that I have learned over the years to try to do this very difficult but necessary task at the same time of building and thinking together. So 1, always coming back to this concept, you have to start with great data. Great data is going to enable you to move fast. Do you know how much easier it is to batch analyze samples that all look like this? These are 6 different samples from the Cellular Immunology Core Lab, run on 6 totally different time points, different months apart. This is a reference control. And all I had to do was draw one gate, apply to all, QC, boom, 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 done. It is so much faster if you start with reproducible data. Number 2, a reminder, once we get to stage 4, we've gone through default settings, we've learned to make UMAPs and P heat maps, come back to the spreadsheet. This is just a reminder that it is okay. It is okay to do basic analyses. It's okay to present basic analyses to your PI, to your colleague, even at your lab meeting. It's okay to use conditional formatting and even horrible box plots and bar plots made in Excel. It's going to actually save you a lot of time. The other thing that is great about going back to basics in the spreadsheet is you start to be forced to format your data in a very structured way. And that also will expedite your analysis once you start getting to the level of automation. I cannot stress enough how much faster your analysis in life can be if you start to learn how to use tools like R to automate your analysis. For example, in Prism, you have to copy and paste to make individual plots one at a time. If you then go back and adjust your gates, you have to recopy and paste. This also introduces errors, and it is quite slow. However, if you work from a structured spreadsheet, you write a code once, and the first time could take you a while, could take you a day, it could take you 2 days. But every time longitudinally you add data to that spreadsheet, you run the same code over and over and over again. And 5 seconds later, you've analyzed every single cell population. Maybe you're looking at 50, 100 different cell populations at the same time with statistics on it. You can just do that repeatedly. It will save you a lot of time. And again, coming back to the concept of reducing your time building so you can spend more time thinking. The less time I am spending copying and pasting into Prism, and the less time I'm spending just pressing the run button, the more time I can do what I want to be doing, which is thinking about the biology. And just coming back to this, just a trick and a tip, if I have persuaded you to use R in automation, you must have perfect data. I'm just telling you now, you cannot have leading or trailing spaces. You can't have an extra dash. You must match cases. The data has to be perfect. Just FYI, there is an upfront learning curve on this. And then the last point here, and I don't see this done very often, but I do think it can make analyses efficient, is if you use high dimensional tools on analyzed data. So if you have a spreadsheet of analyzed cell populations and you run a PCA on it, I know it's not a UMAP, but the difference here is PCA actually gives you an output of which variables contributed to the variance and difference between groups. So instead of having to analyze 100 cell populations, you can run a PCA on your analyzed sample and it will tell you, hey, take a look at these populations that are creating differences between the group. Then I go back and I just plot that population, and lo and behold, there's differences between groups that the PCA told me. This is not on the raw data, it's on the analyzed data, and the output is it will guide you at the populations to look at so you don't have to look at everything. And then the special case of building and thinking at the same time is UMAPs with raw data. I have made so many UMAPs that I do feel like I can speak to this with some authority. They are very hard to do well at scale. You have to have even more perfect data than I have already described. They are very sensitive to batch effects. Also, you have to think about things like proportional downsampling. You should be comparing apples to apples, 100,000 cells from this sample and 100,000 cells from that sample if you're trying to be even remotely quantitative about it. And it may also require parameter processing. Like if you acquired the data under APC-H7 or APC space H7, it is going to give you, it's just very difficult to do. My suggestion is if you are trying to do UMAPs during the thinking and building stage, keep it very simple. Compare conditions within experiments. Just stop worrying about batch effects for as long as you possibly can. And then the second is to use one sample at a time to explore uncharacterized populations. Make sure you're accounting for all the different possible cell types in your data set. But I don't suggest trying to go for the full downsampling of every single cell type, concatenating between experiments. In my experience, it has been more work than it has been worth it in most cases during this building and thinking stage. Now, one way, I just wanted to show you one example of how I might use it during building thinking stage. This is a cytokine stimulation assay. So I could see with my raw UMAP, I was just looking at 2 samples here, responders on the, no stim on the left, I should have labeled that, and stim on the right. I drew a gate around these cell types that to me looked different between samples. I could see that the stim sample on the right had this new cell population. And so I then exported that raw data and I put it into R and I did a heat map just positive and negative. Setting the positive is anything that had a threshold above the manual data that said, for example, interferon gamma was started at 1,000, like 10 to the 3 MFI and higher. I made that red. Black was negative. So this is positive and negative. If you're doing a manual Boolean gating strategy and you have 10 cytokines, there are 1,024 possible populations that you can look at. And you would have to gate all those and look at them over time and over samples. This did take me a couple of hours to do. It might take you an afternoon or a day, but instead of looking at 1,024 populations, which would crash whatever software you're in and not be very useful or efficient, I would then spend the afternoon making this, and I found that there were actually only 21 relevant populations that had 10 or more cells, and all I had to do was then draw 21 gates instead and focus on the actual relevant cell types. So again, another example of more work up front, but less work over time. And I just want to say that this beautiful example came from OMIP 60, one of my favorite OMIPs, and I could download the data from Flow Repository. If people don't know about this resource, go to Flow Repository and practice doing all these different analyses with great data. It can also show you what great data looks like if you're new and you're not quite sure. Okay, now, special case of how to think and build at the same time. I use AI all of the time, every single day. So I felt like it was important to include here because it has made my work much faster and more efficient. These are some ways in which I think you could, if you wanted to, use AI. One, teach me how to use R. I cannot stress more, or Python, if you have a preference. Teach me how to use it. It will walk you step by step, everything you need to download, and you're gonna be up and running in 20 minutes. Number two, write me a code to plot every cell population in my data with statistics. That is what I did to come up with this. Realistically, this is actually the length and description of prompt that you would need to come up with that graph. So a better way of doing it, as somebody who does it all the time, write me a code to analyze my data in this style. Copy and paste the image of the figure that you want, then it knows a lot better than your description typically is. And here, I am not suggesting that you upload your data to the AI. I'm suggesting that you ask it to write a code to generate the plot in that style in like R or another software so that your data is actually never uploaded to AI. It's just imported into your local software, just to be clear. Third, write me a code to run a PCA in R. And four, I am not supporting this. That's why I have a caution symbol here, but I think people are probably doing this. Prompt 4, analyze this data for me. The idea of using AI and all of these suggestions I've made is to reduce your time building so you can spend more time thinking. Do not offload the thinking to AI. It's really not as good as you are. It's just regurgitating existing information and it tells you what you want to hear. Also, it's not safe to do so. A lot of universities are going to be very concerned about you accidentally uploading proprietary information or patient confidentiality information. We have no idea really where the uploads or imports that we're putting into AI are going. I think I already made the second point. You're the thinker now, not the builder. But on that too, if you're asking it to make plots for you, to make statistics for you, you also need to be able to read line by line and ask it, which statistic are we using here? Why is this the appropriate statistic? You're the thinker now, not the builder. So you're not spending all your time making the plots, but you should be spending the time making sure you're using the right statistical test and so on. And then, as we all know, but just a reminder, AI hallucinates. So even if you decide that you want to import your spreadsheet to AI and say, make a figure for me, you have no way of verifying that the points in the figure that it's making for you are accurate. So I suggest using it as an assistant, but not using it to think for you. That's why we're all in biology, because we love to think about this stuff. All right, so third part out of 4. After building and thinking, you're now ready to publish your data. So there are standards for publishing flow cytometry data. Those are not standards that I have come up with. They're standards that the journals require of you. And then I have 2 resources here about standards at the bottom. the original guide for the perplexed in 2006 and an updated guide for the perplexed published in 2021. So I'm going to be going through how to display your flow cytometry data, scaling, plot types, standard requirements, and formatting. So you can go from this default figure on the left to a nicely annotated and scaled figure on the right. So first, bi-exponential scaling is a must. It's a default in most flow cytometry programs. Fun fact, this is the 20th anniversary year of logical, which was the algorithm that enabled us to see negative data in the linear range around 0, and then also positive data in the exponential range. That was really transformative because before that there was just a logarithmic scale and there were a lot of artifacts like this and lots of data in the negative range. So it's already automatically defaulted to bi-exponential, but I thought it was a fun fact to include. So in practice, what you want to do is adjust your scale to maximize the range of the data. In this example here, there's a lot of empty space. The data is compressed. You want to adjust your scale. Instead of maxing out at 10 to the 5, max out at 10 to the 4. The data is better separated, easier to gate. There's less empty space. Next, scale your data to ensure your negative population is unimodally distributed around 0. Here is an example where you have a histogram around 0. And if you start playing with the scales too much, you're going to find that soon that population, that histogram around 0 is going to split in half. And there's going to be nothing around 0. And that is not correct. That is an artifact of scaling and it's incorrect. Don't do this, do this. Next, scaling cannot fix comp and unmixing issues. This is a comp issue, this is a comp issue, this is a comp issue. Scaling is never going to make that go away. That has to go away with proper controls. Now, scaling UMAPs. This is one of my favorite and least favorite topics. In general, UMAPs is just not my favorite topic anymore, but anyway. So in this example, this person had a no stim and stim in the same experiment, same animal cell type, and they scaled the no stim from -0.1 to 1, and the stim was scaled from 0 to 3 on the stim. I actually don't know what units those are. Make sure to put your units on any sort of heat map that you're doing. But the other thing is this is extremely misleading. To scale first of all, you have to scale all your samples the same way. Stem and no stem, condition 1, condition 2, they've got to be scaled the same way, especially like within an experiment where the scales should be absolutely the same. And second of all, you can see how you can use scales to mislead. In the no stem, it looks here like there's actually interferon gamma expression in these red areas, but there is no interferon gamma expression. The red here is 0.1, which corresponds to the blue area here. The other thing I'll just say about this is to match UMAPs as much as you can to compare them. And you do that by concatenating files together. You run the program and then you gate to split out the samples separately. So it's better to do it together than to do things separately. Yeah, I'll also just add that it is not easy to scale heat maps in R. This was probably done at R. In my experience, there's not like an equivalent of a bi-exponential scaling type of thing in R for UMAP. So if somebody has it all figured out, let me know. Okay, another tip. When you're scaling in a default software program, originally your data might look like this. And here I should have labeled this as a CD4 expression on your UMAP. Once you scale, though, to maximize the range of the data, you'll see that this UMAP is now better contrasted. It's orange. That is a tip for how to make your UMAPs look better, just in your default software programs. Another way that I like to scale UMAPs is with the manual thresholds. So for example, here, this is expression of granzyme B in red. I have colored the negative data in white. It's very clear that the positives are red, and you can still tell the difference between the light granzyme B and the dark granzyme B. A tip to do this, export the CSV as scaled, because the flow softwares do a better job of scaling flow data than your Rs and Pythons and so on. And then you start to set your breaks and limits manually. Okay. Next, you should publish your data using pseudocolor or contour plots, plus use big dots for rare events. So this is a pseudocolor plot. The coloring shows that there is more density, more cells in the red areas. When you have rare cells, try to use big dots. And this is an example of a contour plot with big dots. The big dots and the contour plots and pseudocolor, they do a nice job of telling you how many cells are in different parts of your plot and just allowing you to see them all, because they disappear with the small dots. And the reason why we do contour and pseudocolor and not just regular dot plots is because you lose the density when you just use the flat dots without any sort of contour. Okay. This is the full list of standard reporting requirements according to Nature, and I think they're acceptable too. Include a numerical value for cell count or percent. Make sure the plots are pseudocolor contour. Gates are drawn in the software. Don't draw gates in other programs like PowerPoint, because then your statistics might not be accurate. I just think it's a better idea to do it in the software. I've seen it done both ways. Make sure the axes match. This is kind of similar to the UMAP that had different scales. Make sure that the axes in comparing data are exactly the same. And then make sure the axis labels are large and state the marker and floor used. Next, formatting. Oh no. That is so ironic. Oh, here we are. So you want to use Arial, as much as it pains me to say, because Helvetica is and will always be the superior font, and that is why there is a documentary about it, and it's very good for anybody who is a font freak like I am. But anyway, Arial is a safe choice because it is compatible between Macs and PCs. It basically exists in every software program that there is. But if you have the opportunity to use Helvetica in your figures, definitely don't be shy. It will level you up. Technically, the Nature reporting requirements allow you to use size 5 fonts on your plots, but I couldn't bear to put that on something that was going to be on YouTube because it's not very accessible in my opinion. So I would suggest 6+. I just wanted to point out that if you like these fonts, these are all Helvetica, made to look different ways. Now, the other important thing is colors. In my experience, so hard to pick your own colors if you are not a graphic designer or a color theory expert. There are so many palettes available online. All you have to do is find a palette that you like, ideally a colorblind friendly palette, copy and paste it into your program of choice that you're using to make your figures, and then use this little eyedropper tool and click the colors on that palette that you have copied and pasted, and boom, now all your figures are in a beautiful professional palette. People that have made these palettes are gonna be better at it than you. Just go with that. All right, so saving figures in the right format. Default, generally you want to save pictures as PDF. PDF is a vector. Contrast that with PNG, which is a raster. Vector files are infinitely scalable because they're made of paths and connecting points, whereas raster files are pixels on a screen. So as you zoom and scale them up, sometimes you lose resolution. However, vector plots can be larger than PNGs. And so for that reason, UMAPs, which if you're publishing a UMAP that has 500,000 plots on it, in a PDF, it's a vector. So every single one of those points has its own layer, and it will take forever to load it. And your editor is going to say, we can't accept this 1,000 megabyte file. So you want to save very high density plots as high resolution PNGs, 300 DPI, save them large, then you can zoom them in and out. Do not save them small, because then you'll lose resolution as you zoom in. And last, in this part, the toolkit. Hold on, I'm trying to figure out what time. How am I? 2 o'clock. Okay. So using the right tool for the job. There's so many different tools that you can use, and I just want to highlight the ones that I use because I've been doing this for a while. I use any of the flow softwares are fine. I love using R also because it's free. I have my own startup, and if budget is an issue for you in your lab, R can get so much of this done for you for free, and it is more flexible than any of the other softwares. Also has a steeper learning curve, so that's the downside. You can get most of these elements that you need for publishing data done in JMP. Prism is not as good at automation, and I can't speak more highly about Adobe Illustrator, if you're serious about professional quality plots, like R, it's the most flexible platform because it can work with vectors and layers. So you can change and perfect every little part of your plot in a way that you cannot do in PowerPoint. So my recommended workflow: first, check journal requirements, set up your assembly and finishing program, like the layout based on the final print size that the journal says, export your plots from the analysis program, PDFs as a default, PNG for things like UMaps, and then import those into the finishing program, ungroup, unclip, release masks, select and fix fonts, adjust lines, weights, and align. In Adobe, you can actually select all fonts at the same time, change them all at once, change the sizes all at once. It's just very efficient once you figure out how to use it. Specifically to Helvetica, thank you. Yes, always. So, oh, this is a little messed up too. That's okay. This is the last part of the talk. Principles of data visualization, flow cytometry edition. So Edward Tufte is a master of data visualization, and he has this quote: "Simplicity is not the absence of complexity, but the presence of clarity." And the idea is that the figure should disappear. You should immediately be able to look at a figure and figure out what the point is. What am I trying to say to you? And so in this example, this is Kim Kardashian's house, and you can immediately see that the point is to sit in this chair and read this book. There is no ambiguity. So be a Kim. Now, here are some quick tips on how to make your figure disappear so the biology can stand out and appear. 1, reduce variables. Move extra stuff to the supplement. Reduce colors. Colors are also not accessible sometimes. Reduce ink and use borders to sharpen. So in this example, we're including sex, female and male, but there's really no significant difference. So we can eliminate that as a variable here, and maybe we put it into the supplement to compare if somebody is interested. What that does is it also reduces the colors, so now it's in grayscale and it's more accessible. Here I've also deleted the background grids, and now you can clearly see on this picture on the right, the healthy control group has more T cells than both of the other groups. The low dose has, the high dose has the least amount of T cells. It's just immediately clear what the point of this picture on the right is, and you have to then spend very little time on it to understand what it's showing you. These are also 2 great resources here I want to point out. This Visual Strategies for Biological Data is a series that came out in Nature Methods, and it goes through every part of scientific plot design that you could ever want, and it's really beautifully done. I can't speak more highly of it. So now here's an example. If I'm looking at drug A and I have a UMAP and drug B, it's not immediate. I think it looks like the cell type has gone down a little bit. It's not really immediately clear. And here's where, in my opinion, the lowly bar plot wins. It's immediately clear to me that CD8 cells are going down by a significant amount. By comparison, in this example, the CD8 cells are fully depleted. And it actually is pretty intriguing to look at it by UMAP because you can see the other cells are not depleted at the same time. So that tells a different story. So it's not that any one type of plot is better than another. It's about knowing when and how to use them to communicate your message. Now, just a few examples of other principles of data visualization. Here, we can use contrast to make a data point stand out. Here, we use contrast and containment, right? I made these points lighter, these ones darker, and I added a dark square. I'm reducing the cognitive load of the person looking at it. I'm telling you, look here. Here is an example where this is a traditional ICS Boolean type of plot, lots of pluses and minuses here, and I just added my own little twist to it to take away the colors, reduce ink. I'm taking away erroneous minuses. You're only looking at the positives here, and I'm moving the number of functions directly onto the data so you can immediately see that the cells with 1 function are mostly CD8 cells and the cells with 4 functions are mostly CD4. Just these concepts of reducing ink and reducing cognitive load. Now, I told you that most of the time you want to be Kim Kardashian. However, some of the time you want to be Lenny Kravitz. You just have to know when to be him. So fun fact, he is an interior designer. He's a maximalist, and he designs the most beautiful interior designs I think I've ever seen. This is cut off, but sometimes the complexity is in fact the clarity. It is in fact the point. And there are some examples here. In this study, I personally have a very hard time interpreting spice plots. I stare at them for a very long time and I still cannot figure them out. But this is a very elegant way, in my opinion, to use it because you get the main idea. There's a lot going on, and you can see that there's not a lot going on at 2 hours, but over time, there's a lot more going on. So these T cells are doing a lot, and they're doing a bunch of different things. That's the idea. Maybe you support this with some easier plots to actually quantify exactly what it is they're doing in a simple way. Second, in this case, the complexity is the clarity. There's also a lot going on in this plot, but you immediately get the idea. Wow, there are a lot of different cell types. They're related to each other in these different ways. And last, OMIP69. Again, the complexity was the clarity because the whole point was to show how many cells we can see in this one sample type now with 40 marker flow cytometry. So in conclusion, I've introduced the concept of how hard it is to think and build and different strategies to do so at the same time. Start with great data. It's okay to go back to basics, use automation, and use AI as your assistant, but not to think for you. I reviewed the standard reporting requirements for making your figures acceptable for publication, and we went through principles of data visualization. And the goal here is to reach thoughtful communication. So now we know when to use a bar plot and when to be Lenny Kravitz.